Discovering the RNA World with Gerald Joyce by Tyler Farmer, Lindsay Now, Imed Jamai, and Rahul Ramanathan. As we know it, proteins and enzymes are the core of most biological processes. But how did those proteins come about? Could there have been a simpler form of existence before proteins and enzymes? You might remember the Miller-Urey experiment. They simulated the gases in early Earth's atmosphere, and they found that certain amino acids had formed spontaneously from the inorganic gases. Dr. Walter Gilbert in 1986 proposed a world that started out with just RNA, no formed proteins, enzymes, or anything else. So how could have just RNA led to the complex proteins we have today? That's where Dr. Gerald Joyce comes in. Dr. Joyce is a chemical biologist at the Salk Institute. His work there was eventually able to provide evidence for a world that only consisted of RNA. He was eventually able to create a self-replicating polymerase made of RNA, called a ribozyme, that synthesizes different important parts of a functioning genome, like aptamers and even tRNA, all without a single protein. Let's try and understand how he did that. To start off, a ribozyme is like mRNA, but with functional properties. For example, it can have polymerase activity. He engineered a ribozyme like that using a few advantageous properties. He introduced mutations into the molecule so that he had over 10 to the 14th ribozyme variants. He even used in vitro evolution to select for the most efficient ribozyme. They put the ribozymes to test whether they could extend primers that coded for aptamers, which are sites where a ribozyme can bind to. In his quest to find the best polymerase ribozyme, he wanted to refine the generated ribozymes, so he used something called a gel shift procedure. After analyzing the population, he finally found a polymerase ribozyme with 17 mutations that had much better efficiency than the wild type. Joyce named his ribozyme Polymerase 24-3. To test the functionality of Polymerase 24-3, Joyce used a form of PCR called Ribo-PCR to amplify a certain gene. In Ribo-PCR, there's only RNA, so no proteins or enzymes to help out. Using an RNA template, RNA primers, and the 4 NTPs, Polymerase 24-3 was much more efficient than the wild-type ribozyme that had a 40,000-fold amplification of the template RNA in just 24 hours. The synthesis of these RNA structures supports the hypothesis that ribozymes were one of the key molecules to exist in early life, and gives further insight into the chemical origins of DNA.